these mechanisms that we are going to discuss and how to move forward to get tools, reagents, and actually new ideas to see what we can drug and what we can study further. Today, these 25 years of PDK1 and Akt will be actually a lot of inspiration for many of us. For some of you, they might feel like old papers. For some of us, they seem that they are yesterday paper, although 25 years have gone through. With that, I would like to introduce Dario Alessi. Dario was born in France, but as many of the people here, there is a very strong link to the UK and in particularly Dundee. At Dundee, he studied with Philip Cohen and later became a professor of signaling transduction in 2003. And the MRC protein phosphorylation and ubiquitination in 2012. His focus on phospho and ubiquitination has been definitely to design therapeutic approaches as the director of the division of the signal transduction therapeutics. They are, together with Dundee professors and actually 6 pharma companies, have led to a lot of the discoveries that they are today in use. Today he has a lot more than 200 peer-reviewed papers, 96,000 citations, and to cap it, this year he has received the Louis Genet Prize for Medicine. He doesn't need my introduction, and he had definitely give major contributions to diabetes, cancer, and more recently, Parkinson. With that, Dario.
Okay, thank you for the extremely kind invitation, and also thanks to all the other organizers of this meeting. It's a tremendous pleasure to be here. I don't think I've ever been to a meeting with the word PDK1 in the title of the meeting, so I think this is the first after 25 years. And I haven't worked on the PDK1 pathway for the last 10 or 15 years, so apologies if I'm slightly rusty. I went through my slides a few days ago, and none of them would open from back in the year 2000, so I had to remake all the slides using PDFs from papers. So hopefully it's not too much of a jumble, and and if I'm rusty, I apologize. So the story today starts when I was actually, this is in 1994, I was a postdoc in Philip Cohen's lab, and I was actually working on the RAF protein kinase, trying to understand how it was activated. And I'd been doing that for 3 or 4 years, and I hadn't made any progress. And I'm not, and then Philip gave me the opportunity to, maybe change topic. And I was very interested in the work that was going on in Philip's lab at the time and elsewhere in the world on the insulin signaling pathway. So I thought I would maybe try my luck in a different signaling pathway. So. And there's 2 ways to study a signaling pathway. You can start from the top, from the insulin receptor, and work down to the bottom of the pathway. And the other way to study a pathway is to choose the physiological effect of the pathway, namely the way insulin regulates glucose uptake, amino acid uptake, and protein synthesis, and then work back up the pathway. And the hope is that one day the 2 people working from these different strategies will meet up somewhere in the middle. So in 1994 the upstream part of the pathway was really well understood from the work of Ron Kahn, Lou Cantley, Mike Waterfield, and many others, that the insulin stimulated the activation of PI3 kinase. And this happens very quickly in cells. This happens within 30 seconds. And this seemed to be a key mediator of insulin signaling. And then work from the bottom of the pathway, and I'm taking here Philip Cohen's work. This started in the late 1970s and 1980s. This is a very, very elegant work where, they were studying, this work was started by Ed Krebs and Fisher beforehand, the mechanism by which insulin activates glycogen synthesis by activating the enzyme called glycogen synthase. And it's a very interesting mechanism here. Rather than the phosphorylation stimulating the activity of glycogen synthase, it turns out that glycogen synthase is dephosphorylated to become activated. And then Philip had discovered the kinases that phosphorylate and inhibit glycogen synthase, and this was mediated by this kinase called GSK3 that Philip discovered. And and then the way this works, this was revealed by the work of Callum Sutherland and Philip Cohen, in 1993, I think, when when it was discovered that insulin stimulated the inactivation of GSK3. And that mediates and because you have phosphatases, when GSK3 becomes inactivated, you get dephosphorylation and activation of glycogen synthase. And the inactivation of GSK3 occurs within 2 to 5 minutes time period. So and then Darren Cross, working in Philip's lab and in collaboration with Brian Hemmings, made a very critical discovery in 1995 that PI3 kinase triggered the inactivation of GSK3 by inducing the phosphorylation of this GSK3 the kinase at an N-terminal residue. And so this was sort of where the field was when I started to get involved with work in this research area. And then very important work that had also been done by Darren Cross in Philip Cohen's lab had demonstrated that there's actually 3 kinases that can phosphorylate GSK3 at this N-terminal site. But you can inhibit 2 of the 3 by using rapamycin and the MEK activation inhibitor, and you block P70 and P90 risk. And GSK3 still becomes inactivated by insulin. And only when you have PI3 kinase inhibitors you block this. So this is this was really strong evidence that this PKB was the key kinase that phosphorylates and inactivates GSK3. So this is what the problem was. So we were basically getting close to joining up the signaling pathway. You have PIP3 produced within half a minute, and then maybe 1 minute to 2 minutes later, the PKB becomes activated. And the question is, how does PIP3 induce the activation of PKB? So there was lots of people working in this area. So there was one study by Alex Toker and Luke Hantley at the time, that because PKB has a pH domain at its N-terminus, and Mark Lemon and others had just shown that pH domains bind phosphoinositides. That the idea was that the most obvious idea was that PKB could directly bind to PIP3, and that this would activate it. And Luke Hantley and Alex Toker reported in this paper here, that's actually incredibly well cited, that PI345P2, that's the immediate breakdown product of PI345P3, was able to activate AKT directly. So everyone thought that was the mechanism. PI3 kinase made PI3, that was converted to PI34P2, and PI34P2 then bound to AKT and activated it. But we did some experiments to try and confirm Alex Toker's results, and we worked very close. I was very lucky. The next door, you need a lot of luck in life, and the next door lab to Philip's lab was Pete Downs, who worked on the PI3 kinase pathway, and he worked with these 3 amazing chemists who had spent 10 years making PI345P3 and PI34P2, and the assays we used, that we could confirm that, especially PIP3 and to a lower extent 3,4P2, we now know these bind equally, but in this experiment we saw stronger binding to PIP3, and then you take the PIP3 away and it dissociates. But when I did the kinase assays with with PIP3 and all these things, I could never demonstrate that PIP3 could activate AKT in an immunoprecipitate. So the key question then was, well, you know, is there another mechanism by which PKB is activated? And as many kinases are activated by phosphorylation, it was a no-brainer to explore whether or not, you know, activation of PI3 kinase could lead to the phosphorylation of PKB. So to do this experiment, this was actually one of the hardest experiments I think I've ever done. You had to label, we took L6 myotubes, these are muscle cells that grow on the disc, and then you had to label them with 60 millicuries of 32p phosphate. And I did this about 3 times a week for about 6 months. You know, so I broke all records of radioactivity by doing these experiments. And essentially then you label for 3 hours, you IP the AKT, you run the AKT on the gel, you auto-rad the gel, you cut out the 32p labeled AKT, and then you digest it with trypsin. In those days we had no mass spectrometry machines, so you just digest it with trypsin, and then all you could do was run a HPLC trace, and then collect the tubes with where the 32p peaks are luted. But it was a very beautiful result that in every single time we did this experiment, in unstimulated cells you saw no peaks. After insulin stimulation you saw 2 peaks, PK and peak B, and then you added the PI3 kinase inhibitor, you lost the PK and peak B. So we had no idea what these PK and peak B was, and the only technologies we had, these were very low counts. I don't know if I've got counts there, but you're talking 2,000 counts and maybe 1,500 counts, and so the only technologies we had there was to do phosphoamino acid analysis. So we could tell peak 1 had a phosphoserine and peak 2 had a phosphothreonine. And then we could do Edmonds sequencing, where you couple the peptide to the C-terminus and then you knock off the N-terminal residues one by one, and you look for the cycle in which the phosphate's released. So peak B was very favorable because the phosphate was released after a cycle of 8. And I think there was only one serine in AKT that had a cycle bursts 8 residues, a serine 8 residues away from a lysine or arginine residue, which was serine 473. And then peak B had a cycle burst at cycle 1, and I think threonine 308 was the only one that I think met that criteria. So anyway, we thought that these could be 473 and threonine 308, and to check this, what we did was we mutated the threonine 308 site. And so when these experiments now are done with transient transfection experiments, which are much easier, you get much more material, and we collaborated with the Brian Hemmings lab to do these experiments. And here you see when you mutate the threonine 308 site, you lose the threonine 308 peak, and when you mutate the serine 473 sites, you lose the serine 473 sites. There are these 2 other sites, 124 and this very famous site 450, that I'm sure Alexander Newton and Susan Taylor will tell us more about in the turn motif, that was also found. Presumably we didn't see these in the L6 myotubes because we only labeled for 3 hours, and maybe with the endogenous protein the turnover of these sites isn't strong enough to see labeling, whereas in the transient transfection system we were able to see these sites. So this data suggested that PKB was activated, became phosphorylated at these 2 sites following insulin stimulation. And to demonstrate that this was responsible for activating PKB, we again collaborated, with Brian Hemmings' lab, and Mirana and Angelkovic made the constructs for us, where we transfected the 473 alanine on the threonine 308 alanine mutant into 293 cells, and you can see mutating either one of these sites dramatically inhibits the activation of PKB, suggesting that you need to phosphorylate both sites to fully activate the enzyme. And I won't show you the experiments. If you mutate both sites to glutamic acid, you get some constitutive activation. If you mutate the threonine 308 to D, you kill the enzyme. If you mutate the 473 to a D, you actually you still get normal, quite good activation following insulin stimulation. So. And then we also at the time made, I think this was the first phosphor-specific antibody I ever made, at least in the lab, where we tried to make one against threonine 308 and serine 473. The threonine 308 one didn't work, but the 473 did work, and we actually collaborated with Callum Sutherland in Dundee. I'm not sure this experiment was ever published, but we actually, rather than doing things on cells, Callum suggested taking muscle biopsies from people, stimulated plus and minus insulin, and I actually volunteered for this. So one of these samples is mine, and it was jolly painful. I can tell you, a huge chunk of muscle was cut out of your leg before and after the insulin stimulation, and I remember feeling so ill. They must have injected too much insulin to me, because I remember driving back and feeling completely awful, and I had to stop at the corner shop and buy 2 Mars bars, because I assumed they'd given me too much insulin, and I felt better after the 2 Mars bars. But anyway, this was quite nice because it showed that the insulin stimulated the 473 phosphorylase, and this was very early days. I'm not sure Callum actually published this data. Maybe he did, but I couldn't find it when I was looking through my slides yesterday. And then we worked with John Luco in Dundee, who's a very well-known electron microscopist who did some very careful staining to see where PKB was localized in cells, and this data suggested that IGF-1 was stimulating the translocation of PKB to the membranes of cells in a way that was blocked by by Wirtz-Mannin, and if you chopped off the pH domain, you lost this this translocation. And and then Miriana and John Luco and many other groups in the world also demonstrated that with GFP versions that were just coming online, I think in 1996, that you could see translocation of PKB to the membrane after insulin stimulation. And Miriana did a fantastic experiment as well, where she attached a membrane targeting sequence to PKB, and in this experiment you can see that when you artificially put PKB at the membrane, even without insulin, it becomes maximally activated, suggesting that the key was just simply to take PKB to the membrane, and that was sufficient to induce activation. So this is the 1996 model of where we were. PKB went to the membrane, presumably by binding to PIP3, and it became phosphorylated at these 2 sites, but we didn't know what the upstream kinases were. So the key question was to work out what was the kinase, and the most likely explanation, there's only one step, because this happened within 30 seconds. PKB was activated after 60 seconds in the cell. There's only really time for a single kinase here, and we speculated that there might be an upstream kinase that depended on PIP3 in order to phosphorylate PKB, and we did some initial experiments. We took rabbit skeletal muscle extract, and I think we ran them on a Q-cephalose column, and we screened every fraction of the Q-cephalose column for an activity that could phosphorylate PKB. And then we incubated each fraction either just with PCPS vesicles, and that's the open bars, or we incubated them with PC vesicles containing a small mole fraction of PIP3. And you can see in fractions 7 and 8 of this column, there was a huge activity that massively phosphorylated PKB, but only in the presence of the PIP3 vesicles. This was very dramatic observation, and we're also very lucky to be able to do these experiments with Stephen James and Peter Downs and the chemists who could provide us with these lipid mixtures at the time, which was not routine at all. And David will talk next on his work, where he, in parallel to us, also found the PDK1 kinase, and I'll leave David to discuss that. And then we also, just to emphasize that you only see this activation of PKB when you have the PC and PS vesicles. If you add triton to the reaction, you lose the activation. If you have, I think we have PIP3 without, do we have PIP3 without the vesicles? Maybe we don't, but I don't think that works, that doesn't work either. And when we have the vesicles by itself, it didn't work. So, the other key result was this purified PDK1 could, and we call this enzyme the 3-phosphodependent protein kinase for obvious reasons. At the same time, Princess Diana died. And the people in the lab kept on telling me we called it PDK1 for Princess Diana kinase, but I can assure you that that never even occurred to me. But anyway, but the key result was that PDK1 phosphorylates threonine 308, but not serine 473. And then we purified. This was, quite a lot of work at the time. We had to, I think, take sort of 6 or 7 rabbits and take all their skeletal muscle and then do these huge preps where we purified the activity through, I think we did a Q-cephalose column first, and an S-cephalose, a heparin cephalose, and the final mono-S site. And then we pulled the top 3 peaks, and we digested it with trypsin, and we ran out the tryptic peptides on HPLC, and then we did Edmonds sequencing of the peptides to work out what the peptide sequences were. And in those days the human genome wasn't known, so we were very lucky in the lab at the time that we had Maria Diek, Antonio Casamayor, and also Nick Morris did a lot, and Barry Cordwell did a lot of these peptide sequences. These are the peptides that we obtained from the purification, and we found a protein that contained all of these peptides. I should say, key to completing, I think we were able to clone the N-terminal bit, but we could never get the C-terminal bit. And I was very lucky that I met Chris Marshall, who works with Alan Ashworth. So Alan Ashworth's a very famous cancer biologist, and in 1995 he just discovered the BRCA2 gene, which is a big deal. Now he's the head of the cancer center in San Francisco, and everyone said he's the best British molecular biologist. So I faxed him the sequences of these peptides, and literally, I think within a day, or maybe even 2 days, he faxed me back the whole sequence. And he'd been able to assemble. He said he worked day and night for 36 hours or something to get the sequence. But that was, and in parallel, I acknowledge that David Stoker, working with Len Stevens and Phil Hawkins, also did the similar thing, and luckily we found the same proteins. So the sequences suggested that the PDK1 had a kinase domain at its N-terminus and the pH domain at its C-terminus, the opposite way around to PKB. And interestingly, although this wasn't reported in the human genome, there was a paper on a Drosophila kinase called DSTPK61 that had the similar structure and similar sequences, and this was worked by a group in Edinburgh just next to Dundee. Mary Bounds and Diane Harbison worked on this. And interestingly, this is a kinase that was discovered because it was differentially spliced between male and female flies. I still have no idea why PDK1 would need to be differentially spliced in flies, but that was apparently the observation, and so they actually had some protein from this. So they came to Dundee. Diane came to Dundee, and then we were able to assay it, and it confirmed that the recombinant fly protein could phosphorylate and activate PKB, and that was exciting because it showed us that we'd actually purified and cloned the right kinase. Wasn't certain at the time, and then we subsequently cloned the human kinase, and it did the same thing. So the model then was that PDK1 phosphorylates threonine 308, and there was another kinase or another mechanism by which the serine 473 is phosphorylated. We worked so hard on the serine 473 pathway, and I must say this is one area we never made that much progress on, but you'll know the history of this field well. So in 1998, this was the model of how PKB was activated. That is sort of in inactive conformation in the cytosol. When it goes, maybe the threonine 308 can't be accessed by PDK1, maybe the pH domain is blocking it, and when it went to the membrane, you got a conformational change in PKB exposing the threonine 308 and the serine 473 residues, which was become phosphorylated. So the next advance was that then so Ricardo was in the lab, and he worked. He did many projects I'll tell you about, but one of the first ones he did, he worked very hard with my colleague Dan Van Alten to get the crystal structure of the catalytic domain of PDK1. And he also worked, David Commander, who was I think a master student in the lab working with Dan as well, was involved in this project. And then David stayed on to do his PhD in my lab and Dan's lab, and part of his PhD he also got the structure of the pH domain bound to PIP3 and other inositides, and that this was the second paper here. So this is the structure of the kinase domain and pH domain, and this wasn't just a box ticking exercise. I'm going to show you in the remaining slides of my talk that the structural information that was obtained here was absolutely critical for understanding the regulation and function of PDK1 and designing very precise mutations that enabled us to test the models of how the pathway operates. So it was really critical. So around this time as well, in 1998, many other groups were starting to show that other AGC kinases were activated by insulin and other growth factors. I've got PKCs down here, but that's probably not right, because I don't think PI3 kinase really activates many PKCs. But anyway, this is the slide I had in 1998, and obviously it was clear that all of these kinases were activated by T-loop phosphorylation, at a similar site to threonine 308 on PKB. And the question was, could PDK1 be a master upstream kinase? So at this stage there was an explosion of research, and I've probably missed many papers here, but we for P70S6 kinase, it was our group and George Thomas and Brian Hemmings showed that P70S6 kinase was activated. For PKCs, it was Alex Toker and Alexander Newton, and also I think Peter Parker had a paper as well on this to show that PDK1 was upstream. Morten Freuden, who's in the audience, was able to demonstrate that PDK1 was the upstream kinase of P90 risk. And Antonio Casamayor, who's in the audience here, he's done amazing work with Jeremy Thorner in yeast to show that yeasts have SGK homologs, and the yeast PDK1 is critical for activating SGKs. So this was all very beautiful work. A lot of this work, apart from Antonio's work, was all biochemical based. And the other thing I wanted to mention here is that many of these papers in these areas were published in Current Biology, and there's about more than a dozen papers, I think. And the interesting thing, Current Biology recently had a thing of their 25th year anniversary, and they published their top 100 papers in the last 25 years, and unfortunately PDK1 didn't make it. So I think that was a big mistake, because there wasn't a single mention of it in this anniversary issue of Current Biology. So so we wanted to prove that PDK1 really was the upstream kinase in mammalian cells, and this was the work of Michaela Williams. No one in my lab or even in Dundee had made the knockout cell, and we were very lucky. We had an ESL expert in the unit, Valeria Poli, and Michaela was able to work with Valeria, who to generate. We had to use gene conversion to knock out the PDK1, and when you knock out the PDK1, you lose the threonine 308 phosphorylation, but not the 473 phosphorylation, and you completely block the activation of all of PKB, P70, S6 kinase, P90 risk, and I think we did also SGK and probably PKC isoforms. So this provided genetic validation that PDK1 was the upstream kinase. Because Susan Taylor's here, we also looked at PKA. I know there's some papers that Susan's published that PDK1 can activate PKA. At least in these ES cells, and I think other cells we've looked at, in the PDK1 knockout cells, we still saw normal or near normal activation and phosphorylation of the threonine 197 site. So obviously, we can maybe discuss that further later on. So I think our work and that of lots of others in the audience and elsewhere in the world suggested that PDK1 was sort of activating probably 24 of the 60 AGC kinase members, and I think this data has stood the test of time over the last 20 years or so. So now I'm going to come to Ricardo's work, and I must say I do think this was a work of genius that Ricardo did here. He completely single-handedly realized that the mechanism by which this was working. So by looking at a very careful sequence alignment, he noted that these hydrophobic residues and the hydrophobic motif were conserved in PKA, and this motif in PKA bound to a pocket in the kinase domain of PKA, and that was really critical for the activation of PKA. And he termed this pocket the PIF pocket, and it was conserved in PDK1. All other AGC kinases other than PDK1 have a hydrophobic motif, so the PIF pocket is occupied by their own hydrophobic motif. But PDK1 has a PIF pocket, but it has no hydrophobic motif. And Ricardo realized that this would be the docking mechanism by which PDK1 would interact with many of the AGC kinase substrates. And you can see there's a in the crystal structure he obtained, I think this is a sulfate molecule, was bound to a residue next to this famous PIF pocket. And then the other critical thing Ricardo did, he made mutations around this PIF pocket site here, and looked at the impact that they had. So there's a peptide called piftide that comes from the PRK2 hydrophobic motif. And when you add piftide and you're assaying PDK1 here with a peptide encompassing the threonine 308 residue of PKB. So when you add piftide to wild type PDK1, the peptide binds to the PIF pocket, and this stabilizes PDK1, and you get activation, very nice activation. And then he discovered this very famous 155E mutation that actually is it must mimic the peptide bound form, because the enzyme becomes constitutively active and it isn't activated further by adding the PIF peptide. And then Ricardo measured the binding of these different the relative binding of these different hydrophobic motifs in their phosphorylated state to the PDK1 PIF pocket, and he found that obviously the PRK2 sequence bound very tightly, but the S6 kinase and SGK bound more tightly than the PKB sequence that's shown here. And so that was his first paper in this area, and the second paper he realized that basically this would be a phosphate binding site, so that the phosphorylation of the hydrophobic motif would be the key for enabling PDK1 to dock onto these AGC kinases and phosphorylate the T-loop site. And I just want to show one piece of data that came from this paper that Ricardo did. It's a very nice piece of biochemistry. You can see that S6 kinase is phosphorylated by PDK1, but not the PIF binding mutant. The same with the E mutant of S6 kinase. If you have the PDK1 PIF mutant, you can't activate S6 kinase, you can't activate SGK. If you chop off the pH domain of PKB, you actually have an SGK-like enzyme that has no, and that's still activated by PDK1, but now it becomes dependent on the PIF pocket, because if you mutate the PIF pocket, you strongly ablate the activation of the pH-less PKB. But when you do the assays with PKB, you can see these PIF binding mutants are activated normally, suggesting that for PKB you do not need the PIF pocket. For PKB, we think it's the binding of PIP3 and PDK1 at the membrane that's driving the activation of these enzymes. So this is the model that Ricardo came up with, which I think has also stood the test of time. For PKB, this is the mechanism we've discussed. For the other kinases that don't have a PIP3 binding pH domain, the key regulation is the hydrophobic motif, and you've got these 24 kinases that all have to become activated independently from each other, and it's not working by PDK1 becoming activated. The signal works, it selects which of the downstream kinases need to be activated by phosphorylating the hydrophobic motif, and then this then triggers the interaction of PDK1 with the hydrophobic motif and the phosphorylation and activation of these downstream kinases. So anyway, this is a very nice biochemical model. We wanted to formally prove this, and so I think after Ricardo left the lab, a PhD student, Barry Collins, in the lab, he made some knock-ins. These were done in ES cells originally, where he made knock-in ES cells with this mutation, and you can see with PKB, when you have the PIF mutation, you get normal activation of PKB, the same as Ricardo found in the biochemical experiments. But when you look at P70 S6 kinase, I haven't got the data here, the same as with RISC, SGK, and probably PKCs, you get zero activation with the or virtually no activation with the PIF binding mutant. And then Jose Bayascas, who's also in the second row there, did a very beautiful. This is the only paper, I think, in my life that we sent to the journal and it got accepted without any revisions. Is my recollection correct, Jose?
Yeah.
So Jose, because all of the previous experiments were done in ES cells, and then Jose wanted to do this in mouse because he said you can't believe results from ES cells. So he worked out ways to make conditional knock-in mice, where you could knock in skeletal muscle, I believe this was, or in heart muscle, you could introduce these 155E mutations. And here again, in the this was heart muscle, was it, or was skeletal muscle?
It was
Heart muscle. The hearts they're not so fused. It's harder to do these experiments in skeletal muscle where the cells fuse, and you have one cell in 100 that hasn't got a knockout, and the PDK1 diffuses all over the muscle, so the heart muscles are easier for this. And in this experiment here again, you can see the PIF binding mutant, you get activation of PKB, and but when you do, I think this is S6 kinase activity, you can see it's not activated in the PIF binding mocket, and the same with the RISC mutation. And the same with S6 kinase 2. In those days, we assayed both of these enzymes. So, so this was, I think, very convincing. It validated all of Ricardo's findings. And the last bit I'm going to come to now is, does PIP3 activate PDK1? I know this has been a hot topic in the last, a few years or so. And I just want to show you our old data on this. So, I can't remember whether I did this experiment or other people in the lab did this experiment. Probably everyone in my lab who came to my lab did try at one stage or another to see whether PDK1 could be activated by PIP3 in cells, because it was the most obvious experiment to do. And but we could never find, if you take PKB without the pH domain, it's activated by PDK1. But when you add PIP3 under conditions you can activate the full-length enzyme, you see no activation of PDK1. So this is all biochemistry. With peptides, with delta pH PKB, to my recollection, no one in my lab could ever demonstrate that PDK1 was activated directly by binding to PIP3. But to prove it, what we decided to do was to, look at the crystals. Well, we looked at the crystal structure of the pH domain bound to PIP3, and we did two studies. We made, the first study was Ed McManus, and in those days we didn't yet have the crystal structure when we did this work, so we predicted some arginine residues that turned out to be correct, and we made the mutations here, but this mutation slightly reduced expression of PDK1, so it wasn't ideal. But the more elegant study was done by Jose Bayascas, where he mutated this lysine 465 site that binds, I think, to the 3 and the 4 phosphates. It's the really key lysine residue that binds the PIP3. And he mutated this residue to a glutamic acid. And this completely ablates PDK1 binding to PIP3 to 0. So, and then Jose made a knock-in mouse with this mutation. And so the results I'm going to show you are Jose's results, but we got the same results with this other mutation. So we've got the same data basically with 2 different mutants of PDK1 that can't bind PIP3. So if the model is that PIP3 activates PDK1, then one would expect that all PDK1 substrates could not be activated when you introduce this mutation, if that model is correct. So then there was a really key experiment to prove the in vitro experiment, and this is the result that we got. So I think I'm showing you PKB first. So with PKB in skeletal muscle, heart, liver, adipose, ES cells, the PIP3 binding mutation largely inhibits, not completely, the activation of PKB. So it's possible that there's other mechanisms that can take PDK1 to the membrane and enable it to phosphorylate PKB that goes to the membrane. So PKB activation is largely inhibited, which is what we expect, because the model is that PDK1 has to be at the membrane in order to activate PKB. But the key data is here with S6 kinase. So you take the easiest way to ask the S6 kinase is you starve mice and then you feed them, and you measure the activation of S6 kinase after feeding, and you can see that the activation is not affected by the PIP3 binding mutant. And the same thing to measure risk in mice, the easiest way we found was to inject the mice with insulin, and you can see a sort of a twofold activation in the heart. But we did these experiments with ES cells and other cells, and I haven't got time to show you all the data, but basically the results are always the same. If you ablate the ability of PDK1 to bind PIP3 in vivo in mice, you largely block PKB, but you don't really affect S6 kinase or RISC. And I'm not sure whether we did SGK or these. We did SGK. Jose's nodding. So, yeah. So this is my last slide. So I think this is the model we had. I think in the mid 2000s, which I think I don't know whether it's still is correct or not. But for PKB, it's very clear that without insulin, PKB can't be activated. After binding to PIP3, we likely get a major conformational change that enables it to be phosphorylated at threonine threoate. I think there's a lot of data to suggest it's mTORC2 that phosphorylates serine 473, but Alexander Newton has persuasive data that at least for the PKC isoforms that this is all mediated by a complex sort of phosphorylation reaction that also involves mTORC2 in boosting this sort of phosphorylation. And then once this C-terminal site is phosphorylated, it binds to the PIF pocket on the PKB kinase, and this is the mechanism by which PKB is activated. Once this C-terminal site docks, and this was all sort of discovered by Ricardo, and then the activation for these other S6 kinase, SGKs, RISC, and maybe PKCs as well. You get a signal that induces the hydrophobic motif phosphorylation. This doesn't activate the kinases, but enables them to dock onto PDK1, which then phosphorylates the T-loop site, and then the phosphorylation of the hydrophobic motif then, like here, binds to the empty PIF pocket on the kinase domain, thereby activating the kinase. So this is where we were in 2005 or 6. And then I decided to work on other kinases, because but rightly or wrongly, that's where we left it, and I look forward to learning more at this meeting about what progress has been made since 2005 in this field. And I just really want to say that I'm showing here the people in my lab who worked on this project, and you see I've bolded, underlined, amazing. I must say it was incredible. I just started in my lab, and to have such a group of incredible collaborative researchers who all work together and talk to each other in a time. It was quite, amazing, very, very fortunate. And I hope I haven't forgotten anybody. I think these are the key members of the team that did all the work, in my lab and made big contributions. I haven't mentioned Maria Diex, who did all the cloning in my lab and contributed massively to the projects discussed. Margaret Lawler's work I haven't really discussed, but she made some hypomorphic mice that were really important in understanding that you could reduce 90% of PDK1 in mice and you still activated PKB more or less normally, and suggesting that the pathway is very sensitive. And this probably explains why it's hard to use PDK1 inhibitors to block the pathway, because even if you just block 99% of the PDK1, the 1% that's left is enough to activate AKT. So that's why it's very hard to block AKT activation with PDK1 inhibitors. The system is just so efficient. And I think all the others I have, mentioned during my talk. Anun Zoran was my first PhD student, and he was critical in getting things off the ground and discovering this peptide that ended up to be quite useful observation. So I hope I haven't waffled on too long and bored you, but thank you very much for listening to me.
Thank you for this excellent presentation, and with that I would like to see if there are questions from you all.
I have two questions. One, it was very nice to see that hello. It was nice to see that you actually tried to put some timescales for some key signaling there. So the shortest timescale that you identify, I think, was 30 seconds for one phosphorylation. So those timescales, do you think that those are upper limits? I mean, how? Because anyway, I will be talking about timescale tomorrow, and I think that the timescale could be faster than.
Yeah, no. I think this was the way we did experiments. Is you stimulate the cells with a pipette, and then you have to shake them, and then put them back in the incubator, and then rust slice. So 30 seconds is the shortest.
Manual.
But nowadays, obviously, with single cell stuff, you can probably do this in milliseconds, and so. I mean, it would be fantastic to do this more. I'm sure people are doing this. This was just done we're talking 25 years ago.
Yeah.
So, but even though these rough scales for me were very critical for the path, the signal moves along the pathway, down the pathway. So each step takes time to find this next component. So having the time scales was very useful to really understand what was going on.
Absolutely.
I think.
Absolutely. And the second question is, did you identify any phosphate, actually a specific phosphosite that will actually bind to that phosphosite that you clearly show next to the peptide? I mean,
yes, so
you have the motif, but do you have one case in where that
So basically the site I showed you is on PDK1.
Right.
And the phosphate site is critical for binding. I didn't show you that. We had I think we called it the phosphate groove. I might be wrong, but Barry Collins, my student, he actually made a mutation in the site that bound the phosphate, not the 155. So we made a second site whose number I don't recall now.
131.
131. That's right. Arginine 131, and. So that and that's recapitulated all the data.
So that will also block S66K
Yes.
the same way as
Exactly the same way.
so you can mutate the PIF pocket or the phosphosite
yes,
pocket,
very good
and both.
question. Yes,
Okay.
very good question.
All right. Thank you.
Yeah, no, we did that. I should have shown that data, but it's.
Thank you.
Another question.
Is this working? Yeah. Thank you, Dario. It was a lovely trip down memory lane. Just on the last few slides about whether PDK1 needs PIP3, I should know this, but has anyone looked at the naturally occurring P10 mutants in prostate cancer or GBM, where there's a higher, a longer half-life of PIP3? you know, that might be an informative way to see whether there's sort of pathophysiological activation or extra activation of
To cross, you
PDK1.
mean, so we could have crossed these knock-in mice with the P10 mice to see whether they get tumors or something? Is that what you're saying?
Or just look at the P10 knockout cell lines and assess PDK1 activity.
I don't think we did this experiment, but I think we didn't cross the 155E mice, did we? With the P10 mice, that would have been. So we crossed the P10 mice with the PDK1 hypermorphic mouse, and that's reduced cancers. And but we didn't think to try that. I don't think at the time, but that would be very interesting to try that, because yeah.
Dario, that was a really nice trip into the history. I wanted to ask you about all of the reported PDK1 substrates that don't have a membrane binding domain. So
Yes.
they all have a hydrophobic motif and they all have a PIF pocket, which means that they bind that hydrophobic motif in cis, and this often regulates their activity. How do you imagine that PDK1, which would require trans interaction with that hydrophobic motif, how do you imagine that those kinases are that lack a membrane binding domain and that would allow a conformational change to expose that hydrophobic motif and perhaps also
So
lead
what
to its
you're saying is
phosphorylation?
that maybe when you hit this motif, this would immediately bind to here and not be available for PDK1
Right, exactly.
docking.
There's
Is that your
basically an
question?
intramolecular competition
Yes.
that would dominate unless you could locally concentrate your substrate with
Yes.
PDK1. Now that's easy to explain, for example, with AKT, PKB in the context of the membrane, but it's not so easy to explain
So,
with some
so
of the
your
other
question is
substrates.
whether when this motif here, why it doesn't just bind to here
Yep.
and not be available for PDK1 to dock.
Right.
Okay, so obviously we don't know. Maybe that pocket isn't. I'm just guessing now. Maybe the structural biologists would be able to confirm this. But one hypothesis would be that the pocket that this binds to is not properly formed till you phosphorylate the hydrophobic motif. And Ricardo's nodding, I see. So basically, this can't bind here because the pocket's not made, and the pocket only is properly made once you phosphorylate this site. That's just my prediction. But Ricardo's nodding, so maybe we should talk to Ricardo about that.
Susan has a point to it.
So I'm thinking maybe it's a C-terminal tail, which I'll also talk about that. Maybe in this case, the C-terminal tail isn't really latched on top of the end lobe of the kinase until you do, in this case here, until you do the phosphorylation of the activation loop, because it's got to bind. If you don't even have, without the C-terminal tail wrapped around there, you don't even have a proper way for ATP to bind to PDK1 or to. I don't know.
So you're suggesting that the phosphorylation of this site helps make the pocket.
Helps make it helps wrap the C-terminal tail around the whole end lobe, and once it's wrapped around, then you have the.
So that explains that would explain why PDK1 would be able to recognize
Right.
whilst it's in an unformed state, then it
Right.
would dock. That would be the guess. But I haven't looked into this.
I
so.
haven't really thought about the cis-trans aspect of it, but I think that might explain it. Yeah.
Is a follow-up or?
No, it's not. But,
I want to ask.
go ahead.
Great talk, Dario. I just wanted to follow up on your question. So in the case of PKC, although it does bind a phosphorylated hydrophobic, PDK1 prefers to bind a phosphorylated hydrophobic motif, in the context of the cell, it binds an unphosphorylated C-terminal tail. So if you do co-immunoprecipitation experiments, newly synthesized PKC that's got no phosphates anywhere binds PDK1 very, very tightly.
Yeah, but it's possible because the phosphate might just enhance the binding,
Exactly.
but the
And
PIF
wha-
pocket is still able, I think, to bind. Is that possible, Ricardo? The non-phosphorylated. It's just maybe stronger with the phosphorylated,
Exactly.
but it's not.
Exactly.
It's not
So-
all or none. It's not.
Exactly. So in the case of PKC, although it has a higher affinity for phosphate at that site, once PKC is phosphorylated, that hydrophobic motif site is no longer accessible to PDK1. So it's actually binding mechanistically and never phosphorylated. PKC does the activation loop which then causes the hydrophobic motif to bind its own PIF pocket. Yeah.
Yeah, no, that seems reasonable. Yes, yeah.
Thomas.
This is maybe a heretical question.
Yes.
It's just out of curiosity, but you mentioned at the very beginning that when you were thinking about this time window, there was space for one upstream kinase.
Yes.
I'm just wondering, at the time, did you consider the possibility of autophosphorylation, or was that very easy to rule out?
No, I think, well, we couldn't. Well, we knew that I think we did try to incubate AKT or PKB with PIP3, and I mean, I'm not sure. We couldn't see activation. All I know is that we probably didn't do peptide mapping to look at this, but we did do sort of in vitro experiments, and in those days we didn't have very good recombinant. We made GST PKB I think in 1997, and that was helpful. But in our hands, if you just incubated the PKB with PIP3, you didn't see any phosphorylation of threonine 308. Under some situations, when you have PIP3 and very large amounts of the PIF-tide peptide, we could get some phosphorylation of serine 473. But everyone told me this was just a physiological artifact, so we left it at that. But then I've come back to look at some of Alexander Newton's data that he might discuss, and it does seem theoretically possible that the 473 might involve an element of autophosphor. I mean, we'll let Alexander tell us more about that. So I still think the 473, we first found MAPCAP kinase 2 as the 473 kinase. We proved that wasn't the case. Then we had this and we proved that wasn't the case. And then David Sabatini came with the mTOR story that looked quite good. The strange thing with if you IP the TORC, the TORC2 complex, and you incubate it with AKT, you can see but you see so little phosphorylation. Well, you had 1/100th of the amount of MAPCAP kinase 2, that's the non-physiological kinase. You see 1 mole per mole, whereas you're seeing like 0.1 mole per mole with mTORC2.
I mean, at least in our hands, purified TORC2 doesn't do anything to AKT. On 473. We can't see any signal.
Yeah. So, I mean, I'm sure Jose Bayascas and others in the room will remember how hard we tried to find this PDK2. I had several grants, which we never succeeded in doing anything because we could never make any progress here. So yeah.
So I think.